So I'm going to be doing my presentation on Leonard Adleman. His work kind of interfaced with biomedical engineering, which is my major, and so that's why I chose him. So first of all, his developments. One of his first developments was an algorithm to distinguish prime numbers from composite numbers, which he used later on in some of his other work as well. And then he's most well known for RSA cryptosystem, which we'll talk a little bit more about in the upcoming slides. And then he also was one of the kind of first people to start looking into DNA computing, which has been an up and coming way to store data, and just a lot of cool stuff happening in that field right now. And he kind of started that whole thing. So first, RSA cryptology. RSA stands for Ravesh Shamir Adleman, who are all the founders of it. And it's a method of security known as encryption, and it's actually the most widely used method of encryption. And in 1985, Leonard Adleman and his partners began actually making RSA computer chips to encrypt things within your computer system. And then they actually, he went on to receive a Nobel Prize for computer science because of this technology. And so how does it work? So it encodes information by using what's known as public and private key pairs. And so if you have a message that you want encrypted, you don't want everyone to know, like a password of some type. The message is first encrypted with the public key. And then if, whoever is receiving that data, instead of just having them have that same key, they actually have a separate private key to decrypt the data. And so this prevents both parties from needing a shared key and thus keeps things more secure. You can also mark the data with digital signatures. So if a hacker was to come in and try and access the data, there could be this digital signature on the data which would prohibit them from being able to decode it. And so only the person with the private key would understand how to decode this digital signature, and then thus only the intended audience would receive the data. So it's a very secure way to encrypt data, and it's often used in a lot of different password settings. And like in our own everyday life all the time, banking is a huge one. So credit card transactions all use RSA technology to encrypt your data and keep you more secure. Securing emails often use RSA cryptology as well. And then finally, authenticating phone calls. This also uses RSA. So we see this this type of cryptology in a lot of our everyday life, and it's really made our world more secure in those areas. So then moving on, the second thing that Leonard Adleman is known for is DNA computing. And so DNA computing takes essentially DNA and uses it as data to work as like its own computer and store information. And so DNA itself, for a little bit of background, is made up of 4 nucleotides: adenine, thymine, cytosine, and guanine. And all of these nucleotides can be put into different sequences to get different DNA sequences, and within our own bodies, these DNA sequences make up who we are. They're responsible for telling our body how to produce certain cells, or certain hair colors, eye colors. All these things are found within our DNA. And so in DNA computing, it's a similar idea where these different sequences of these nucleotides can be used as data points to store things. And so naturally, DNA has specific pairing. A pairs to T and C pairs to G. And these pairs allow for DNA computing to have its own logic operations, and or statements kind of type of thing within your data storage, which really makes for a multifaceted programming experience within this computer interface. So there's a lot of advantages to DNA computing. Just in the past year, there's been a lot of advances in it, and people really seem excited about it. And one of the reasons for that is that, as I just mentioned, it allows for multiple operations to be computed at once. Because you have these different levels, you first just have the levels of the pairing of the sequence, but then you also beyond that have pairing happening. You can have multiple operations kind of being computed at once, making for a faster computing system. It also, because of this, also increases speed and efficiency, so you can get through a lot more data a lot quicker without using as much power and that type of stuff. And then also there's the ability to solve complex problems. And this is because of benefits to optimization, and this has been seen in cryptography, pattern recognition, optimization. And so because of the increased speed and efficiency and this multiple operations being computed at once, these more complex problems that maybe take multiple things happening at once can be solved, and thus have really advanced certain fields. And then the other advantage of DNA is it has a very, very large storage density. One singular DNA strand can store about 1 billion gigabytes of data, which is much larger than our current storage processes. And so this would allow us to store large sets of data, and then also be able to solve complex problems using that data, because if you can store more data, you have more information and you can do more with that information. So as everything, there are disadvantages to DNA computing, and that first one is going to be the cost. It's very costly to work with DNA because it's a biological process. There's a lot of lab stuff that goes into it, but then there's also some regulations with it, and so it's very costly. The second thing is that there's a lot of complexity when you start to manipulate things at a biochemical level. Very small changes can actually affect your DNA very drastically, and so that's something that you have to be very careful of when working with it. And not just anyone knows how to fix something created by DNA computing. It takes somebody who understands biology and even chemistry to a certain degree to be able to work with this type of stuff. So that adds to the cost and then just the time it takes to work with this. And then also reliability and accuracy. Biological processes are prone to errors and changes and adaptations, mutations, that type of stuff. And so you do have to worry about those occurring and then thus messing with your data. So overall, I just kind of wanted to present a little bit of a timeline of where DNA computing started and then where it's headed. So in 1994, as mentioned, Leonard Adleman kind of introduced DNA computing. He was kind of the father of this whole thing. And then in 1999, DNA-based logic gates, those and OR gates, started to be developed using that pairing that I talked about earlier. And then jump all the way up to 2012, and we started to see people look into DNA for data storage. And like I mentioned before, DNA has a lot more larger data storage, has a larger density that allows for more data storage. And then in 2016, there started to be parallel DNA computing where you can do multiple operations at once within DNA computing. And then 2020, getting a little closer, more recent, you started to see kind of some hybrid DNA and electronic computing, where people are kind of combining the two because of the limitations in both to get the best result possible. And then 2022, we see and even now we're starting to see emerging applications in fields like biomedical engineering, and there's even talk of the first DNA computer, and if that will take place in our future. So yeah, that is all I have for you guys today. I hope you enjoyed this and thank you for listening.